Pan doktor Altmann jsem ukazoval na jeho nedávnou publikaci z International Archives, člověk, který je biochemik a měl by rozumět biochemické struktuře alergenů, i když se trošičku zdráhal mluvit před lékaři, ale uvidíme, jako biochemik nám zase může říct informace, kterých my si nejsme vědomi. So, stage is yours.
So, thank you. I very much appreciate the invitation of Radek to this wonderful city and to this meeting. And I hope as a non-allergologist, actually as a biochemist specializing on carbohydrates and glycoproteins, I can tell you a few things which are interesting. So the talk will be in two parts. First of all, I will talk about the proteins which are allergens, and you will see a few pictures which are much less interesting than the ones you've seen before. And the second part will focus on glycoproteins, the role the carbohydrate part of many allergens has in allergy, and especially in allergy diagnosis. So this is a picture from my friend and colleague, Wolfgang Hemmer from Vienna, and it shows the prevalence of allergies in Austria. May not be correctly written, excuse me. And you see the most important players are birch, grass and birch. And everything else is much less abundant. Maybe mugwort is important and ragweed is coming. And here you see also a thing of rape seed pollen, which might not really be a true allergen in its own right. Okay, so I can go quickly through the next pictures because you've seen a lot of these things. These are the most relevant grass species, Timothy grass, the Phleum pratense and many others. And there are outliers like maize, which is also not as related to these grass species. So I don't think I have to show this, you've seen that. And interestingly, this is one important point, you have lots of Gramineae families and species, and those who are regularly and importantly allergenic all belong to this family Pylové, to just a few species, and maybe also rye and much less wheat and barley play a role here. So this is so there are many species, only a few play important roles as allergens. From the many leaf trees, it's mainly birch and its close relatives, elder, hornbeam, hazel, maybe also oak, which is also quite related. And this is, so I don't have to show this. And then we come to the third large and important class of allergens, and this is Artemisia and its relatives. It's mainly Artemisia vulgaris, which plays a role, a so-called mugwort, and you've seen much nicer pictures around here. And it's among the wheat pollens, it's the most important allergens. A lot more, approximately 20% of pollen allergic patients react against mugwort. There's another tree, which is ash, which is not related to the Fagales family. It is, say, a real allergy in its own right. Sometimes we find isolated sensitizations just against ash pollen. And then the next important, quantitatively important group is already ragweed, with the probably most important member, the short ragweed. And it's actually an immigrant which originally came to Hungary, and from there, as you've seen already, it is now constantly spreading towards the rest of Europe, and it may soon be one of the most important allergens in our area. It's related to mugwort, but many people really obviously developed a ragweed allergy, even though a large percentage of the patients is cross-sensitized. So at least in immunoplots or in diagnostic tests we find double sensitization with mugwort and ragweed. And then, of course, there are many other things. Nettle, which is maybe related to the Parietaria judaica pollen in the Mediterranean area, of course not important here. Plantain, which again is a close relative to olive, and olive pollen is important again in the southern European area. And there are many other things, special cases, and very often the question arises for the diagnostic person whether such things like strawberry or raspberry allergies or things are really relevant, or just cross reactions which we have in diagnostic tests, whether these things are really relevant as clinical allergies. In Central Europe, I hope this is right, we don't have large problems with gymnosperm pollens. This is different in Southern Europe, where cypresses are also important. The problem in allergy, when I came as a foreigner into this field, is already one of nomenclature. What is an allergen? For many people, the pollen is an allergen, and for other people, the protein from this pollen is the allergen. This already causes some confusion. Maybe we just should call this allergen source, and the protein itself, which reacts with the immune system, should be called allergen. How can we discover the proteins which are reacting with our immune system, of course with the IgE in the end? We extract the pollen, and in this case it has been done by people in Salzburg mainly, in Austria. And first of all, we see when we extract the pollen, we get many, many protein bands, and many of them are totally irrelevant, and some of them bind IgE. That means they could be responsible for the allergic symptoms. And here you see that for this ragweed, many of these allergens have been isolated, cloned, and recombinantly expressed in the hope that these proteins actually are the relevant players. Another approach is inhibition, and what we see here is an immune plot. First of all, we make again an extract of the allergen. We have here the protein separated by size, and we incubate this plot with patient serum. And we see here in an elder extract, patient 1 recognizing 2 allergens, here in another patient in oak pollen extract, other proteins. So this seems simple, but we don't understand it yet. When we make the whole thing patient 1 with oak and patient 2 with elder, it becomes a little bit more complicated, but basically you can forget these things. I'm not going to explain this. And the question here is, are these patients really sensitized against elder on the one hand and against oak on the other hand, or is this, can we simplify this a little bit or understand it? And for this, to understand it better, these plots were inhibited. So in this lane, the serum was, the patient's serum was incubated with pure BET-V1, which is the major birch pollen allergen. And as you can see, this pre-incubation abolished this band. So, and the same thing happened, actually, it's not seen as good, in the oak. Something's wrong with this thing now. Probably, yeah. All right, thank you. So the same thing happens in the oak pollen. So obviously we have 3 different species of pollen, but one molecule is responsible for the reaction here. So the BET-V1 inhibits the elder band here and the quercus, the oak band there. And we can play also with BET-V2. It's just the other band which is here inhibited, and also in the oak pollen. So we have many species, many pollens, many bands, but actually we can now simplify the whole picture a little bit. Because these species are very related taxonomically, also their proteins are very homologous. So when we align these, the BET-V1 and the homologous hazelnut, alnus, hornbeam, etc. Allergens, we see all the red letters. That means here we have total identity. The blue letters we have similarity, and the black letters are different. But what we see, mainly the whole picture is red. That means we have a very high similarity, first of all in the protein sequence, but actually also as a consequence in three-dimensional structure of the protein. And not always, but very often, this means the epitopes binding antibodies are more or less similar. Yeah? In this case, this picture shows us the three-dimensional structure of BET-V1, and the black area here is the area which is recognized by a certain antibody. Yeah? And you see that almost the same binding area can be found on a related allergen from cherry pollen. No, excuse me, not cherry pollen. This is a birch pollen, and this is just cherries, the fruits. Yeah? So there's a relation between birch pollen allergy and sensitization against, which causes oral symptoms, against fruits like cherries, peaches or apples, even soy milk in some patients. And you see here, okay, what you find easily by anamnesis or skin testing, and there's actually a structural basis when you look at these allergens. Even within one species, an allergen might occur in slight variations. For example, the BET-V1, which was of course very thoroughly studied because it's a very important allergen, occurs in many isoforms, and it's not so important to know all of them, but one point might be interesting. Some of them are more allergenic and some are less allergenic, and we speak here of hypoallergenic isoforms, and that might be very interesting in treatment of allergies by specific immune therapy. Sometimes this hypoallergenicity is boosted by altering the structure of the allergen, for example by polymerization or by chemical modification to make these things even more hypoallergenic to assist in the treatment of patients by immune therapy. These are just pictures to show you the structural similarity of BET-V1 with the related allergens, in this case actually the apple fruits and also the hazelnut, which could either occur in the pollen or in the fruit. So we've seen just all these BET-V1 and ALM1 and so. And here, if we go to BET-V2 and look at the homologues of BET-V2, the thing becomes more problematic. So the usual allergen nomenclature gives us no clue that FLI-P12 and MALDI4 and whatever are actually homologues of BET-V2, but they are. It becomes only obvious when we look at the nature of this allergen. It's actually a so-called profilin, is a constituent of the cytoskeleton, and it occurs very ubiquitously. It occurs in all plant cells and actually also in animal cells. Of course, with a slightly different sequence and structure. Again, if we align the sequences, the amino acid sequences of various profilins, we see that the red areas dominate very much, and therefore we have a wide cross-reactivity. This picture shows you an immune plot, so again the extract of tomatoes probed with the sera of various patients. And you see a few patients who react with like E1, and this is said because it's the number one. It should be the major tomato allergen. Which has to be proven, I guess. And the question is this reactivity because these people are really tomato allergic, or because they are simply cross-reacting with the very homologous profiling from tomato? Actually, they were sensitized, or they are allergic against birch pollen or mugwort pollen or whatever. So this is a very difficult question. In the case of these high molecular mass allergens, quote-unquote allergens, yeah, you see a number of bands in the high molecular mass region, and this is almost certain that this reactivity does not really point at allergy in the true sense. It's a cross-reaction because of carbohydrates. This will soon be dealt with in my second part. So this leads also to the already to the problem of discriminating between sensitization, that is the presence of IgE antibodies binding to a certain protein, which we then call allergen, and a real allergy in the sense that this protein really elicits clinical symptoms. And this is actually very difficult. The sensitization is measured in a merely quantitative way by either RAST, EAST, CAP, or however these things are called by the various companies. Or you can also measure it in a component resolved way, that you can look at individual allergens by immunoplot, or by, in a more modern fashion, by allergen arrays. And I think RAST and CAP are done on a very large basis, and sometimes you see that the results do not really fit with those from more biological tests like skin prick test or anamnesis, what is the patient really reactive to, and this I think is a big problem. One way, no, let's say, we think that we have, I should say something, sorry. The difference between these things is hard to explain, but it lies probably in these two items. The one thing is the affinity of the antibody, and the second thing might be that not each IgE binding really causes cross-linking of the IgE on the mast cell, which is the prerequisite for clinical symptoms. All these tests are absolutely happy if you have a monovalent interaction. Yeah, the IgE binds to the allergen just on one point, and that gives you a color on in your RAST and on the immunoplot. But this is not enough for an allergic reaction. So it's polyvalency and probably also affinity, and therefore we have very often the danger of false positive results. And here you see lots of, this is another point. If you go to a list of allergens, you find lots and lots and lots of names of species and pollen types. And this becomes even more complicated if you look at each individual species in the allergen database or the allergen nomenclature website, and you find for each of these grasses many entries depending upon the importance of the allergen. For, of course, Phleum, an important allergen, you have several entries, it's well studied. And so you see, okay, you have the FLEPI1, which is an expansin, medium molecular mass, and others, the FLEPI5, which is now known, I think, to be a ribonuclease, and many others. You flip it, our profilin again. And so the whole thing becomes extremely complicated. then if you look at the importance, the clinical importance, you see that only a few of these allergens are recognized by many of the patients. Yeah, of course, overlapping percentages. Some of these allergens are high molecular mass proteins with glycosylation. You will soon learn that this can be a problem. And the good thing is, for diagnosis, that many of these allergenic grass species have very similar allergens. So again, the Pylové, etc. group are, you see, red all over, so they are very similar. It's almost the same with the Pylové group, which is the second important thing. And so, although the thing of, at first, this matter looks very complicated, it melts down a little bit because we can diagnose these grass pollen allergies by taking actually just 2 different allergens from one single species, and then we will catch more than 90% of the grass pollen allergic patients. So with bringing some system into the whole thing simplifies it at a certain point. So this is just a point about nomenclature. Again, if we compare Pylové with these things, they are the same proteins. If we go to BAT-V1, it's obviously a totally different protein. So this old nomenclature doesn't help us very much. This brings me to the wheat pollen allergens, where again we see ART-V1 is not at all equivalent to EMP-A1, totally different proteins. Interestingly, the homologue of ART-V1 in ragweed has not yet been identified, but I may say so much here that actually there is a homologue and it might be of some importance. So maybe the important things are not yet really discovered in both these weeds. And you see again here the profilins, they show up. You see it's a new group, we have not discussed this yet, the lipid transfer proteins, which are in both species, and also the polcalcins, calcium binding allergens, which also are a frequently observed class of allergens. And that brings me, you can find hundreds and thousands of different allergens by the old nomenclature, but actually if you try to understand what is the biological function of all these allergens, you find out that from the thousands of families, in another paper the number was even larger, we find only 29 protein families, folding families, which contain allergens. And actually from these 29 families, just a few are really important and occur regularly. For example, these pathogenesis-related proteins like BAT-V1, expanses like the grass group 1 allergens, then the profilins, polcalcins, and lipid transfer proteins. There's an interesting story about lipid transfer proteins. When you look at people who have problems when they eat peaches, or especially peach, we find that the culprit protein is PR10, a BAT-V1 relative. Yeah, so it's probably a food allergy which comes into existence because these people originally were sensitized by birch pollen. In the Mediterranean area, however, we see a totally different allergen profile. The people are not reacting with this protein, but with lipid transfer protein. And LTP, this is just a speculation, LTP is the major allergen in Parvietaria, which is an important allergen in the Mediterranean area. So this might be either a genuine peach the prunus fruit allergy or a cross-reaction with pyrethria. So there are a few other proteins, and how much time do you still have? You have an idea? 10 minutes, 5 minutes? No minutes? Okay. So, and this is yet another point. If you get an immune plot where you have reactivity in the high molecular region, this indicates that something's wrong. Yeah. Again, this is a tomato extract. A patient's reacting with allergens here, and if we incubate the serum of the patient with a glycopeptide produced from bromelain, this plant glycoprotein which we cut into small pieces to destroy the protein, and then these peptides are coupled to BSA, so we have, and if we use this as an inhibitor, we can completely inhibit the reactivity of this patient with tomato extract. So the conclusion is that the reaction is actually towards the carbohydrate of these allergens, and the patient, in fact, is not tomato allergic. We can do the same thing with other examples, and this is, for example, very important. It's not a pollen, but it's wasp venom in this case. And in this patient, we see that the inhibition completely abolishes the reactivity with the patient's serum. So if a patient has antibodies against these carbohydrates, he will recognize them everywhere, and you tell him that he or her, that she's insect sting allergic, and actually this is not the case. I will not explain the other picture. So we'll use the last minutes to introduce you to the glycobiology of allergens. This is the protein, and it can be actually equipped with sugars. And in a mammal, the sugars look like this. We have a common core which occurs in all eukaryotic cells, from fungi to man. And then we have additional sugars, a very high diversity, which, for example, determines if an influenza virus chooses man or birds as a host. And when we compare these mammalian structures with plants, we see there's a large difference. Yeah? We lack these sialic acids, and we have, on the belly, I could say, two new residues, xylose and fucose. And as these residues are foreign to the mammalian immune system, they are immunogenic. Yeah? So all plant glycoproteins may elicit an immune response in mammals, and then you get a reaction. And we find these glycans on various allergens. Again, we see our FLIPI1. I don't go through the details. Yeah, the peanut allergen, hazelnut allergens, and this is a little bit less important. Ole-E1 from olive pollen. I can skip this one; it's not so important. We even find these types of fucoses in insect venoms, also in parasites, and the question is, what is the role of these sugars? If I have 2 more minutes or 3 more minutes, I could briefly explain that. In the former years, we had some hard time to convince immunologists, especially allergologists, that carbohydrate moieties can really be epitopes. They just said, okay, these are actually peptide epitopes which are just somehow influenced by the presence of the sugar. Actually, what is now accepted is that these carbohydrates are epitopes by themselves, and we can show this by creating these epitopes on a human protein. So what we take human transferrin, the recombinant fucosyl or xylosyl transferases, and we convert the human transferrin to kind of a plant glycoprotein. And if we use then this artificial plant glycoprotein and a control, with patients' sera, and here we have two panels. One panel of sera where we have a very narrow sensitization against only birch or only ash or similar things, and here a very broad sensitization, so-called polyvalent cross-reactive sera. And you can easily see that this polyvalent group, many of these sera strongly react and specifically react with our plantified human transferrin. So with the fucose makes the difference. The gray bar is without fucose, the red ones are with fucose. And we can see this as well with human IgE on an immune plot where you see with fucose binding, without fucose no binding. So I can skip this, it's not so important. The important point is that approximately 20% of allergic patients have IgE against such carbohydrate structures. And that means that this IgE reacts with every glycoprotein from whichever source, from every pollen, from every foodstuff, even with insect venoms in a very, very broad cross-reactive way. That means you get lots of false positive diagnostic results in vitro testings, rust, cup, whatever. The good thing is that it's almost certain that these carbohydrates do not elicit clinical symptoms. Yeah? Clinical symptoms are, if they ever can be observed, the exception. So CCDs, cross-reactive carbohydrate determinants, I can go to the next slide, cross-reactive carbohydrate determinants are a very important reason for irrelevant in vitro results in serum diagnosis. This is certainly the truth, and for allergy diagnosis, this is probably an important point which has to be resolved in the near future by taking into account these glycoprotein reactivities. For the basic researcher, the question is, and this is my last point now, the question is, not the last slide, excuse me, why is this the case? Yeah? For allergologists, it's clear. We have the important protein epitopes and we have the unimportant carbohydrate epitopes. But coming from the carbohydrate side, this is not at all clear, yeah? Because carbohydrates can be specific and wonderful determinants. That's quite clear. So one answer could be that we have a valency problem. Yeah? Many of our glycoallergens are, in fact, monovalent regarding to the glycan. So we have just one oligosaccharide on the allergen, like in ARA-H1, etc. Yeah? But many others are actually polyvalent, like, for example, the horseradish peroxidase, which is not an allergen, but it's frequently used in the laboratory. You probably all have used HRP at some point in your chemical career. And this HRP also is incapable of eliciting clinical symptoms. So valency probably is not the explanation. It could be affinity, yeah? Low affinity, which makes the whole thing ineffective. But what we did during the last month, we purified human anti-CCD IgE and measured the affinity by surface plasmon resonance against fucosylated and here also xylosylated transferrin, so modified human glycoproteins. And we found that the binding constants are actually very, very low. So the binding is, it couldn't be much stronger. So this is exactly where we find other allergens. We also measured the affinity of IgG. You always have, if you have IgE against something, you always have also some IgG in the blood, usually much more actually. And the interesting thing is, okay, this is what I always say. The interesting thing is if we compare our glycan epitopes with real allergens, yeah, FLEB5 or BET-V1, we see that the IgE affinity is approximately of the same magnitude. But if you look at the IgG, and if we put these things into relation, yeah, we see that we have actually a much closer ratio in the glycan epitopes than in the real allergens. Yeah, you see 2 to 6,000, just more or less 1 or 2 orders of magnitude there. So maybe the reason for the low clinical significance of carbohydrates lies in the high affinity of the IgG. And why is this the case? This is, as most people assume, the mechanism by which specific immune therapy works. Yeah? You confront a patient with high doses of the allergen in a controlled environment, and thereby the IgG matures to high affinity. This is also what beekeepers experience who are stung very frequently. They have IgE, but at the same time so much IgG of high affinity that they are immune against allergies. And our hypothesis, which is open to discussion, is that our daily contact, our frequent contact with lots of plant glycoproteins in various ways, mainly as food, but maybe also in other ways, is kind of a specific immune therapy, a sublingual immune therapy, which leads to the ineffectiveness of carbohydrate allergens. I think my time is over, right? Maybe then a last thing about mugwort allergy, which very clearly shows you the difference between proteins and natural glycoproteins. If we look at a recombinant ARTV1, the major mugwort pollen allergen, it's just one nice protein thing. The natural stuff looks like this. It's very, very different. And actually, again here, and I will not go into detail, we have a certain type of glycosylation which plays a role in allergenicity of this mugwort. But I don't have to go into all these details. It's a rather specialized case. And the last thing I want to do is thank all my collaborators, thank especially Wolfgang Hemmer, who gave me some clues about pollen allergens, and you, the auditory, for your attention. Thank you.
I have a question. Who is changing, human beings or the nature? Why do we have more and more allergies? Is it because...
Is this true?
I don't know.
The question, is this true? Do we have more and more allergies?
My opinion is that the number of allergic people grows. Also the audience can confirm it or not. Je pravda, že počet alergiků je čím dál větší a větší? Máte stejnou zkušenost nebo ne? Yeah. So they also confirm that the number of allergic people increases. A moje otázka je, jestli se měníme my, anebo jestli se mění příroda, whether we change or the nature changes because we grow various plants in increased concentration. Also we eat more food than what we naturally would eat. So we are exposed to stronger concentration. And also my other question is that I think that on one part nature changes, but we also change because that's, for example, this food allergy symptom that when you eat apple, it's digested by saliva and you lose the allergenic potential in the stomach. And what we do, we clean our teeth, so we destroy natural bacteria which normally would be responsible for toxic, maybe acid environment in our mouth that would cleave the potential allergen. Also we use various anti-acidity drug for the stomach, so we also lower the acid concentration of our stomach environment, and this may result in stronger allergenicity in our body. And if it's true that we have like 7 groups of proteins which are responsible, maybe more, I don't know how many you listed, which have
29 according to
29. So and they are through all plants and food products. So it means that we are in high danger because we are for sure exposed to all of them. So we will have more and more allergies almost to everything if it's everywhere. So we have to do something, not with the nature, but probably with us, because we started to respond to those 29 potential allergens. Out of biochemical point of view, if I eat apple, what happens with the apple? I have some kind of acidity in my mouth, enzymes in the stomach. If it enters my body, the glycoproteins, in which part the glycoproteins are cleaved? In the mouth? Or?
I mean, the idea is that the glycoproteins, the glycans don't matter so much. This is the idea, yeah. But the allergens are, I think, cleaved in the stomach, not in the mouth. So the atmosphere is not very acidic, and most people say we don't have a lot of proteases in the saliva. It's amylases, but so the cleavage takes place in. But there's one point maybe, and this is important. Many people like this hygiene hypothesis, which is interesting because in the Eastern Germany, the incidence of asthma allergy in children was much lower than in Western Germany, although the environmental pollution was much worse. Yeah, excuse me, you know, and the things started to change when the habits of the mothers in Eastern Germany also changed. They stayed at home much more. The children were not given so much to the kindergartens as before. And suddenly they developed more asthma and allergy because they stayed at home. The conditions were much more hygienic, and the immune system was challenged too little, obviously. So it was bored and developed allergies. That's, I don't know, but this is one hypothesis. You probably know.
Thank you. Máš někdo otázku nějakou? Já bych teď thank you very much. Thank you. I think we will return back to also biochemistry when we talk about immunology. Že ještě budeme potom spojovat tu biochemii s paní doktorkou Agdis a ještě předtím by byl pan profesor Barták, který by nás byl takový úvod do imunologie a potom tím si trošku odpočineme od angličtiny a pak bychom poprosili paní doktorku Agdis. Before you give the lecture, Professor Barták will give some kind of introduction in T-cell, antigen-presenting cell field, and then you switch. So also the audience has a chance to hear the Czech language and then you switch to English again. Pane profesore, moc.